Hey again, Year 10. So this is a quick recap of proteins. So again, this is in the PowerPoint that you can download. I just want to talk you through a little bit of information that I would like you to remember by the end of this week. So it's reminding you of some of the key ideas from last term, and the paper booklet that we gave out has got a lot of this information in it. Okay. So firstly, the monomer of the protein is an amino acid. So amino acids have a consistent structure. They all have an amino group or an amine group. They all have a carboxyl group. They all have a central carbon. The only difference between the 20 amino acids that we know of is this little R. Now that R can be a range of different atoms, and that's where the difference occurs in your amino acids. Now the amino acid is the monomer of all proteins. They join together to form a polymer, and that polymer is called a polypeptide chain. So remembering that monomer is a single building block and a polymer is a long chain of monomers, the terms that you need to know are amino acids are the monomers, and the polymers, that long beaded chain as represented down here, is called a polypeptide. Again, these monomers join together by joining an amino group to the carboxyl group of the next amino acid, and then the amino group joins to the next carboxyl group. So they join in a linear fashion. And again, the same chemical reaction is the one that joins these monomers to produce a polymer. So the term or the reaction that we learned in nucleic acids applies here as well. Condensation because it releases a water molecule, and polymerization because it's building a polymer. Again, it releases water and it requires ATP. So later on this term, we'll start to talk about different types of proteins and enzymes as proteins that help biochemical reactions to occur more effectively or efficiently. Proteins have a number of different structures, so the reality is that there are very complex, long, multiple chains of polypeptides all bound together. And here we have a diagram of hemoglobin, which is a protein that is carrying oxygen around in your blood right now. But before we get to this complex structure, it's important that you remember that it starts off with a chain of amino acids, like a beaded necklace. And each bead is different. Each bead represents one different amino acid, and these little three-letter codes here represent different amino acids all joined together. So we have methionine and phenylalanine and asparagine and alanine and arginine. All of these, you don't need the names, but you need to know that they're all joined in a particular order, and that order is determined by the DNA or the gene that coded for the protein. So the primary structure is that simple chain of amino acids in whatever order it might be. Once that chain forms, and this happens at the ribosome, it will fold into pleated sheets that we call beta-pleated sheets and little spirals, and it will do that spontaneously. So it will start to form more complex structures and then start to fold in on itself to form a three-dimensional shape. The secondary structure is the pleats and the coils, and the tertiary structure is when that all starts to form a three-dimensional shape that will eventually determine the protein's function. And then the final structure, which we call the quaternary structure, which doesn't always exist. Some proteins are too simple to have a quaternary structure. But the quaternary means that there are multiple polypeptide chains that actually come together to form that final protein. Okay, so review this if you feel you need to. Otherwise, move on to your class notebook and start answering me some questions.